Hello everyone and welcome back to our training series on protein model quality. Previously in the series, we explored how AlphaFold's predicted aligned error metric, shortened as PAE, can be imported and interpreted in Jalview. Today, we will consider some more functions related to this metric and how these can help us get a better idea about the presence and relative positions of domains within a protein's predicted structure. Let's work with the calmodulin alignment featured in the last video, consisting of sequences from 5 species: yeast, social amoeba, roundworm, cow, and us, humans. Now, the 3D beacon search we conducted in the previous video revealed AlphaFold models for each of these proteins, including various metrics associated with them from the AlphaFold database. These included the predicted local distance difference test score, shown in Jalview as the AlphaFold reliability score, the secondary structure, and the predicted aligned error, or PAE. And these were all loaded as reference tracks into the Jalview environment at the bottom. These are specific to each sequence in the alignment where AlphaFold has data, and the ordering above reflects the ordering below. For the purposes of this video, I will focus on the calmodulin sequence from yeast, this one up here. Now, if I right-click on the PAE annotations label and select the cluster matrix option, Jalview clusters the columns of the PAE matrix to produce a dendrogram like this. This can be partitioned by cutting it like this. Notice how the clusters change? Group colors are randomly assigned and can be mapped onto the annotation track, the alignment, as well as the structure loaded in the 3D viewer. And changing the partitioning of the tree changes the corresponding colors. But how are the group colors actually mapped onto the various views? By simply right-clicking on the PAE matrix's annotation label and choosing Color by Annotation per sequence. Once this box opens, just enable the Use Original Colors tick box, so Jalview knows which color scheme you wish to use. And that maps the group colors onto the alignment as well as the AlphaFold structure I have loaded here in ChimeraX. Now let's take a look at partitioning the dendrogram. Partitioning it at this point shows the tree splitting into 2 main groups, corresponding to these 2 segments of the PAE plot. This gives us additional evidence that there are 2 major domains of the multi-domain protein positioned in space. So Jalview's cluster analysis demonstrates that what we see in the PAE visualization is indeed reflected in the clustering. Changing the partitioning to this point reveals something new concerning this segment that connects the 2 domains. This segment is also highlighted, you'll notice, up here in the alignment window in the amino acid sequence, as well as here in the 3D viewer. Pro tip: if you lose sight of the tree as you move back and forth between Jalview's various windows, don't forget to retrieve it from the Windows menu up here. Finally, unticking the Show Groups on Matrix option will remove the coloring from the PAE annotation track entirely. And that's it for today. If you found this video useful, don't forget to like and subscribe to our YouTube channel, where we have new videos coming up for all your training needs. Take care and happy analyzing!